Welcome everyone, and thanks for joining us today for this series of webinars on how electron diffraction is transforming research across the fields of materials, life science, and chemistry. My name is Fraser White, and I'll be your host today. It's my pleasure to introduce today's presenter, Dr. Simon Bates, who's our Vice President of Science and Technology at Rigaku Americas. Simon is a highly experienced person in diffraction methods, using everything from X-rays to neutrons and now electrons. Simon has experience in both instrument development and contract research for pharmaceutical materials, so he's well qualified to give this presentation. This is the fourth webinar in the series of 6, and today's topic is the role of electron diffraction in pharmaceutical research. Before we get to the main presentation, I just wanted to inform you of a few quick items. So as always, if you have a question you'd like to ask, please do so using the Q&A button, and these will be read out and answered as time allows. Any questions that we don't have time to answer, we'll respond to after the session. And if you can, please try to include the slide number so that if we get further through the presentation, it helps us understand the context. The chat is open, but please note that if you ask any questions in there, we might miss them as we won't be expecting to see them there. And as more messages come in, they may be buried in the history. We will be recording this webinar, and the recording will be available from tomorrow. If you have any trouble watching the presentation live, we recommend catching up via the recording, as it's unlikely that we'll be able to resolve any technical problems during the live webinar. And lastly, if you haven't already, we encourage you to register for the whole series of webinars on the XtaLAB Synergy-ED. You can do that using the link which I'm posting into the chat right now. Even if you've registered for the individual episodes, you can still register for the whole series. So thank you for your patience, and I'll now pass over to Simon. So Simon, please begin when you're ready.
Sure. Thank you, Fraser. Thanks for the introduction. Greetings, everybody. Welcome. And thank you all for joining our webinar today about solving pharma's toughest solid form challenges with electron diffraction. In the webinar, we'll be talking about where electron diffraction excels in the drug discovery and development pipeline. Special considerations for electron diffraction. Before we start doing our measurements, there are certain things that we really need to get a handle on for electron diffraction. Then sample handling, and for me, which is really the heart of electron diffraction in the industrial environment, is automation, integrated workflows, and multi-grain analysis. Well, then we'll touch on to some routine applications in the pharma space, things that are being performed on pretty much a daily basis now. And then towards the end of the presentation, I'll very quickly show some data on some new areas that we're looking into, like lipid nanoparticles, disordered systems, and total diffraction. So where does electron diffraction excel in the drug discovery and development pipeline? Well, I hope after you've listened to this webinar that you'll be convinced, like I am, that electron diffraction has a wide range of applicability over the drug discovery and development pipeline, all the way from discovery through automated protein screening, looking at ligands, through to studying the drug product itself, looking at tablets, looking at the API form in the tablet, looking at counterfeits, and reverse engineering. So what I'd like to do is step through each of these regions one at a time, and let's see where electron diffraction can be applied. So the first area I'd like to look at is in the development pre-formulation phase. That's where the bulk of the polymorph screening will be typically carried out. And for those of you who are familiar with electron diffraction, obviously polymorph screening is one of the first things that would leap out as a good application area. Now, some of the advantages of electron diffraction is it uses sub-micrograms of as-made powder. First of all, yes, I view electron diffraction as a powder technique. It's a powder diffractometer. It just measures the powder grain by grain. And it looks at sub-micrograms, even nanograms of material. That's extremely small amounts of material. And what I mean by as-made powder is that when you first get solids crashing out in discovery, early development, that should be enough material to put into an electron diffractometer and start doing polymorph screening. You certainly do not need to do any scale-up at all. So you save all the scale-up time and you can start screening immediately as first solids come out. Now, if the impurity profile is too high, you may be just getting oils, or you may not even be getting anything that looks like a crystal. So you may have to do a purification step, but that's not always the case. As soon as you get solids that have some crystals in them, you can put them into the electron diffractometer and start doing polymorph screening very, very quickly. Now, another one of the unique aspects of electron diffraction, and this is often missed, is that the polymorph screening and characterization is essentially unit cell based. So if you contrast that to X-ray diffraction, X-ray powder diffraction, or Raman or something like that, these are spectra based. You have a whole stream of data, a powder pattern, a Raman spectra, and you're comparing one data set with another data set. And based upon the differences or the sameness, you're deciding, do I have the same polymorph or is it perhaps a different polymorph? And then the question, of course, comes up, well, how different does the data have to be in order for it to be considered a different polymorph? And often it's not clear. And you could have made a mixture and that you then go ahead and call it a new polymorph, but in actual fact, it's a mixture of other forms. Now, in electron diffraction, because this is a unit cell-based characterization and searching, as soon as you've got the unit cell, you've uniquely characterized that particular polymorph. If you get a different unit cell, then it's clearly a different polymorph. For those of us who have sort of forgotten in dim history what a unit cell is, I have this schematic picture on the left-hand side here. This is a 2D representation of the structure of ICE-1H, one of the many polymorphs of ICE. And the green drawing in the center here, that represents the unit cell. Each unit cell has 6 degrees of freedom. So that's A, B, C, they're the lengths, and alpha, beta, gamma are the angles. And depending on the ratios between those 6 parameters, it falls into one of the Bravais lattices, with cubic with a single variable, and triclinic up there with 6 variables. And once we've identified A, B, C, alpha, beta, gamma for that particular unit cell, we have uniquely characterized that crystalline form or that polymorphic entity. Furthermore, electron diffraction is rather sensitive to hydrogens. You'd have to go through the dynamic calculation pathway in order to really properly place the hydrogens. But electron diffraction is uniquely sensitive to hydrogens, which when you have a crystal with a coformer inside the crystal, we can identify clearly whether you're dealing with a salt or you're dealing with a co-crystal. That is the proton, is the hydrogen exchanged from the API to the coformer, or does it remain attached to the API? And the way that the agency deals with salts and co-crystals rather differently, then it's important to be able to differentiate the materials that you're making. Do you have a salt? Do you have a co-crystal? And one of the things that can be surprising for electron diffraction, it actually has a unique and wide-ranging choice of different in situ environmental stages. So this allows you to explore a wide energy range when you're doing your polymorph screening. You can do low temperatures, high temperatures, different liquid and gas environments, and the such like. And we'll talk a little bit more about that later as we go on through. So now if we move much earlier on in the pipeline, we're in the discovery section. And basically, the way that electron diffraction actually operates lends itself very easily towards automation and high throughput workflows. And automation and high throughput workflows fit very well in discovery and early development, where you're screening through multiple thousands of candidates a week. So the way electron diffraction operates is we have a grid. Usually these are teeny weeny grids about 3 millimeters across. And onto that, we'll spread some grains of material, and then we'll automatically step through aligning the grains. And the first thing that we'll try and do is to discern whether the grains actually diffract. Do they give rise to any diffraction at all? If they diffract, often for some high throughput experiments, that's enough. You now know that this is crystalline material, and your particular experiment has generated crystalline material. If it diffracts, then we can collect more data and we can automatically determine the unit cell. And then if needs be, we can go on from that and derive the molecular connectivity. So it's this 3-level testing, grain by grain. Does it diffract? Is it crystalline? What's the unit cell? Is it a new one? Is it an old one? And then move on to the connectivity of the molecule. Is this the right molecule? Is this the API I should be looking at? Or is it some contaminant that's fallen from the ceiling or something like that? Now, I've mentioned putting on grains and particles and solids and the things like that. But this high throughput automation is also very well suited to liquid-based automation. In fact, we can pipette onto these grids picolitres of material, so we can actually lay out a 360 well, 96 well plate directly onto the grid. Each one is a pipette of picoliters of material taken from the high throughput liquid-based screen that you may be operating. The liquid evaporates typically very quickly, and that lays down solid onto the grid that we can then step through in the same manner to do our characterization. Now, another benefit that this is a unit cell-based technique is that we can match directly to crystal structure prediction. I mean, I'd say most large pharma nowadays have an automated CSP pipeline. As soon as they have a target molecule, you can then calculate multiple millions of potential crystal structures for that molecule. And that can be really beneficial in trying to understand when you've made the most stable solid form. Now, over the years, people have refined those quantum force fields so that it gets more accurate predictions of what is the most stable crystalline form, which is often the one that's most important to initially release in the drug product, so you can be sure that it's not going to change during the manufacturing process or storage. But another approach to this is to do very early polymorph screening, collect structures of multiple polymorphs as soon as you can, and then calibrate the force fields you're using in your CSP to the actual structures you're measuring at the early stage. And again, electron diffraction is very well suited to this. At the early stage, you have a lot more impurity profiles. Those impurity profiles act like random fields. They give you a lot more metastable crystalline forms that you may never see again downstream after you've gone through purification. So this is an incredibly important contribution that electron diffraction makes to the whole pipeline and can shorten the development and make it a lot more confident that you found the most stable solid form. Now, certainly in Christus Pro, we've implemented powder pattern export for all of our electron diffraction data. So every grain we step through, we collect the full diffraction pattern. We can export that as a powder pattern in order to support other workflows, traditional workflows like Rietveld, identification, cluster tools, and the such like. And one of the things, although I haven't done this myself, some of my colleagues do this relatively routinely, is ligand screening using high throughput protein electron diffraction. So yes, we can use electron diffraction to solve protein structures and screen for ligands attaching at particular target sites. That is absolutely possible. So let's move upstream a little bit more. Now we're into the formulation space. And one of the utilities of electron diffraction in formulation is materials characterization. And nowadays, with this huge move towards nanopharmaceutics and nano delivery systems, that really fits in nicely with electron diffraction in that we can image individual nanoparticles and do a diffraction studies and characterization of individual nanoparticles. So this little image on the left-hand side that I popped up, I'll show this again right at the end of the webinar. This is in diffraction mode, so we call it a rough image of lipid nanoparticles. These are pegylated liposome with an API that's crystallized in the middle of it. So we can isolate single nanoparticles and then do electron diffraction grain by grain and try and understand what's going on with those lipid nanoparticles. Another area of materials characterization is the evaluation of disorder. When we have an API, typically we're going to reduce the particle size to improve flow and manufacturability. And that size reduction, taking us down to the micron or even submicron particle size, introduces a lot of disorder into the API. But how do you study that disorder? How do you know what you're doing to the API structure? Well, electron diffraction can help you there. You can isolate the submicron size or near micron size individual particles and do electron diffraction and characterize the disorder, the atomic disorder that's induced through the milling and size reduction process. And something we recently started to look at, in fact, I was doing these measurements today in our lab in Tokyo, and this is an analysis of amorphous solid dispersions. Can we really see with electron diffraction when the API begins to phase separate out of the carrying polymer, when we're starting to get that early nucleation? So again, nanometer size nuclei forming inside the polymers. So this is rather a new area, but these are all applications that electron diffraction can serve very well. And then moving on right to the end to the final drug product, the tablet form, the solid dosage form, let's say. Then for tablets, we can render them down into a powder, put them on a grid, and we can step through again, grain by grain, powder diffraction, grain by grain, and we can then collect enough information to solve the connectivity. For here, for example, we have two API forms that were found within a particular tablet and an excipient. So we can step through grain by grain and completely characterize the crystalline materials. What are the API polymorphs? What are the crystalline APIs in there? So in essence, this is very good at reverse engineering of dosage forms, looking for contaminants, as we mentioned, IP protection, and many other applications in the final drug product.
Okay, so for our first question for the poll, which I've just launched so you can start answering, is we'd like to know which part of the pharma development pipeline you're working in. So the options are on your screen. You have discovery, development, and pre-formulation, formulation, manufacturing, QC, or other. And if you have an other, then please feel free to tell us in the chat. I'll just let this run for a minute and then we can see what the results are, but it looks fairly even so far.
Yeah, it does. That's interesting, yeah. The manufacturing guys are a bit underrepresented.
Yeah, I'll let it run till we get to about 60% participation, I think. And some people are saying not in pharma.
Well, you're still welcome, of course.
Okay, I think I'll end it there. So it looks like at the moment we have discovery as the leading and other, I'm guessing not in pharma being the follow-up.
Yeah, that's very interesting. Thanks. So, as I mentioned in the introduction, when you're doing electron diffraction, there are many like special considerations that you need to take into account before you do your experiment. And the first one is that electrons interact very strongly with matter, some 10 to the 5 to 10 to the 6 times stronger than X-rays. And one of the limitations or advantages that gives us is that we can only look at really, really teeny weeny small samples. And for organic materials, typical pharma materials, then a thickness of 300, 400 nanometers is pretty much the max. Now, the lateral extent can be a little bit bigger than that. We can go up into the microns laterally. But because the sample is rocked during the measurement, we don't want the sample to be too extended, because then as you tilt to like 50 degrees or so, then the electrons are also going to have an oblique pathway through the sample, and it might be too far. Now, on the thin side, so what is the thinnest sample we can look at? Typically, we say around about 30 nanometers or thereabouts. So that should give you enough material to give a pretty good diffraction signal. So that gives an order of magnitude of range from 30 nanometers to 300 or so nanometers as a good size, and that's on the thickness. So the lateral extent can be smaller or larger than that, sort of as needed. Now, another consideration for electron diffraction is it has an extremely short wavelength. So for 200 keV electrons, which is what we actually operate with, we have 200 keV column, the wavelength is about 0.025 angstroms. And that's like 65 times smaller than copper X-rays, which is like 1.54 angstroms. So what that gives us is a very flat Avold sphere. So you're probably thinking, what on earth is a flat Avold sphere, and what does that have to do with anything? Well, in this schematic on the left-hand side, I've shown a 2D cross-section of reciprocal space. This is a reciprocal lattice. All these diffraction peaks, these represent Bragg peaks. And then for an incident electron beam shown in red here, the Avold sphere corresponding to that because of the wavelength is shown in black. And it's almost a straight line. And that passes through multiple Bragg peaks once you're on zone or you're in a diffraction condition. And that means all of these Bragg peaks can scatter at the same time. So this is a multi-beam dynamical case, which is extremely strong dynamical interaction. Now, this dotted circle in the middle here, that's actually for molybdenum X-rays. So you can see from molybdenum X-rays on that same reciprocal space lattice, it only typically intersects one Bragg spot at a time. So this is single diffraction events at a time. So far less probabilities of leading to dynamical effects. And we'll consider the dynamical implications later on, but that is certainly something that you need to think about when you're doing electron diffraction. And the final one is that clearly it's going to require a vacuum beam path. The electron column is going to be under high vacuum, so the sample, unless you do something about it, it's going to be sitting under vacuum. So if you have a very sensitive organic material, you've got volatiles in there, you'll need to put it in a special environment. My recommendation is that pretty much all organics you're working with, you should consider a cryo environment, and maybe even something where they contain gas or liquid cell. And I'll show you some examples of that as we go through. So I wasn't sure when I put this presentation together that I really wanted to get into any technical details, but as dynamical diffraction comes up all the time when you talk about electron diffraction, if you go to your colleagues and you say, oh, look, I'd like to do some electron diffraction, what do you think? Pretty much the first response is going to be, well, you know, you got these dynamical effects, you're going to have troubles sorting things out. But in reality, it turns out that the single crystal structure determination pipeline is really incredibly robust, and you don't typically need exact intensities in order to achieve connectivity. And I think, as I mentioned, we do the screening at these three levels. So our first level is, does it diffract? And you certainly don't need to worry about intensities at that stage. You're just looking for peaks. Do you see peaks? If so, it's crystalline, end of story. The second thing is unit cells. Unit cells are based on peak positions, not peak intensities. So it doesn't matter whether it's dynamic or something else, makes no difference at all for unit cell determination. Now for space group determination, it does make a difference. And for connectivity, it turns out in most cases for relatively small molecules, you just need to sort your reflections into those reflections which are sort of strong and those reflections which are sort of weak. So dynamical sort of interaction with determining connectivity is not so huge. Having said that, if you're going into doing absolute structure, or you want to place electrons, you want to place hydrogens and protons very, very accurately, then you do need to go through a dynamic pipeline. And in Crystallis Pro, we've enabled two different dynamic pipelines, so that if you have to do that, then we have all the support to do the dynamical corrections and dynamical modeling, for example, on absolute structure determination. But for most of the screening and work that we do is absolutely, it doesn't really matter one way or the other. On this schematic on the left-hand side, I've really tried to indicate, you know, what do I mean by dynamical diffraction and how does it happen and what do you need for it to occur? Well, we have this incoming electron beam in blue, and here's a idealized perfect crystal in the Bragg and Lowy model with perfect atomic planes. As the electron beam shoots through, it strongly interacts with these atomic planes, and the electrons are diffracted out at another angle. And of course, they hit another plane in a perfect crystal, they're diffracted in, and so on. You get this complex beam path through the sample itself. And this looks like essentially an interferometer, which is exactly what it is. And this requires a highly perfect crystals, strong alignment of the crystal planes, and you have to be exactly on zone axis to set up this complex interference effect. Now, most of the particles that we will study, most of the grains we look at, are very, very small, and they are somewhat disordered. We're going to be looking at early stage materials, probably kinetic crash out, going to be not equilibrium grown crystals, poor quality, or we're going to be looking at highly milled materials of the API. So most of the crystals that we'll be looking at are highly disordered, and when you have disordered crystals, you break up these lattice planes, you do not have dynamical diffraction at all. There's not enough perfection to establish this interference effect. Now, if you look in a little bit more detail as exactly when dynamical diffraction occurs, if I take my crystal and I rock it through the Bragg peak, so we have our samples and we rock them through the diffraction condition. As we start to get diffraction, the intensity of the Bragg peak will begin to increase up, and it'll just increase in the usual way, and according to kinematic diffraction, until it reaches this critical threshold of interaction with the material. And once that happens, you'll then hit this interferometer situation, and most of the electrons that are supposed to pass into the diffracted beam can be completely lost within the sample itself. So this gives a flat top peak, which is called a Darwin curve. And it's only in this range of the Darwin width itself here that you're getting a dynamical diffraction. And most Darwin widths are like 0.002 degrees. They're extraordinarily narrow. So you can introduce very clever data collection methods like the rocking of the sample that as you scan across the material itself, 99.9% of the time it's entirely kinematic diffraction. Rarely do you get under these high diffraction conditions on zone axis where you hit the dynamical peak. So again, because of the way we collect data, because we're looking at these small disordered materials, we rarely enter into strong dynamical effects. If they do occur and you need to do absolute structure determination, then we have the pipeline to solve that. And it turns out that dynamical effects are actually really useful if you are looking at absolute structure, placing hydrogens, as I mentioned, but also local structure evaluation, like total diffraction. So to have this interferometer effect is really a strong local probe of exactly what's going on inside the material. So it can actually be a useful effect as well. But for me, the most critical aspect of electron diffraction to enable it to be used in an industrial environment is this integrated workflow from introducing the sample, identifying the grains that you want to look at, automatically stepping through aligning, checking that they're crystalline. If they're crystalline, collect enough data to get the index solution, to get the unit cells, then sorting on the unit cells, combining data if necessary, and then going into connectivity. So this integrated workflow to me really is the most critical aspect to electron diffraction. And this is the heart, in my mind, of applying it into an industrial environment. If you have to do everything by hand, it can be painful. But once it's all automated and integrated, it's relatively straightforward to collect the data and to do the analysis. So sample handling. As I mentioned, because we're in a vacuum, a very high vacuum environment, we really have to be careful about how we handle the materials. And typically, the powders are laid down on a TEM grid. These are about 3 millimeters across. And each of these TEM grids has an electron transparent substrate material. The one I usually use a carbon-based, amorphous carbon, but you can use polymers, graphene, and more recently I've seen a lot of discussion about silicon nitride backing polymers. So the grains are like dusted on top of the grid and they stick to the backing polymer so you can do your measurement. And there are many examples of substrates. The standard one is just flat carbon, let's say, somewhere between 3 and 20 nanometers in thickness typically. But with their flat substrate, if you have like a well-defined habit in your crystals, let's say they're all plates, when you drop them on top of the holder, they can all line up on the polymer. And then if you do diffraction from multiple grains and you want to add the diffraction from multiple grains together, you're essentially double counting because all the grains are going to be aligned in the same way. So it's not really going to help that very much for you to achieve completeness. And so for those situations, we have lacy carbon. This has an undulating surface. So when you drop the grains on there, they're going to be all oriented higgledy-piggledy in different directions. So then when you do diffraction from multiple grains, you can get a more complete data set. And the ones which I typically use, because my focus is more on total diffraction and disordered systems, is wholly carbon. So this has holes in the film. So when I drop the grains on there, a lot of the grains will be across the holes, and so I can shoot right the way through and get a total diffraction pattern without bringing in too much of the substrate material into the measurement. And something I'm seeing more frequently nowadays are these double layer films, particularly graphene. You get double layer of graphene. You can then put the crystals with even some small amount of liquid environment underneath the graphene and seal them together. And some people have had quite some success in doing that. As I mentioned earlier, we can also fit into a liquid-based automation process. If you're looking at natural product isolation or crystallization out of different solvent systems, you have a 96 well plate, 360 well plate, or whatever you have. Using a micropipetter, we can take out liquid from these well plates, deposit them on the grid in another well plate, basically, And then typically, because these are only picoliter size, the solvents evaporate very rapidly. They're depositing the solids. You can then plunge freeze that if needs be, put it on a special transfer, or just drop it straight into a cryo stage and then put that in the electron diffraction system. There's really quite a range of different sample environments that you can choose to do your measurements under. Of course, vacuum comes free of charge because it's a vacuum column. The sample will definitely be under vacuum unless you do something about it. And I definitely recommend a cryo stage when you're working with any pharmaceutical materials because it protects volatiles and protects the material against the electron beam. Many cryostages come with heating capabilities too, so you can go from liquid nitrogen to beyond ambient conditions, in fact, studying a broad range of temperatures and how that's going to impact the crystal polymorphs that are formed. But we also have gas environments and liquid cells as well, which might be surprising to some people, and those can be phenomenally useful. My colleagues in our lab in Frankfurt, in Germany, have been doing a lot of measurements using liquid cell, these nanochannel holders. So we have some mother liquor in there, and we're looking at the actual nucleation inside the liquid itself. So because we can look at diffraction from nanometer-sized crystals, we're looking at the formation of protocrystals, initial nuclei. We can observe the whole Ostwald's rule of stages as it cascades through multiple metastable polymorphs. And just think of the huge energy space that opens up for your polymorph screening. Now you're getting all the early metastable forms that are part of the crystallization and nucleation process. And there's no other way that you can get access to that information. And then you can feed that back to your CSP and refine the quantum force fields and get a very accurate prediction of what actually is the most stable form for this material. It's very powerful capabilities. So at that point, we could move on to a question, Fraser, if anybody
Yeah,
has
we've got
asked any questions.
yeah, we've got quite a few. We've got eight
Oh
questions.
boy.
So I suggest we take a few of them and then move on, and we can pick up the rest after.
All right.
So the first question was slide 10. In terms of polymorph screening, how does this apply for unknown crystal structures?
Sure. But as I mentioned, it's unit cell based. And as you go through grain by grain, you measure you determine the unit cell, and that unit cell is then added to like a hierarchical clustering approach, where you do a cluster tool, where you look at how many of the unit cells that you've measured are different and how many are the same. So it's a sameness and difference study. So even if it's a completely new material, no one has ever looked at it before, there's no information out there, you generate that material during your study. You're going to collect thousands of grains, and those thousands of grains are going to give you thousands of unit cells, hopefully. And then you can cluster those unit cells between the ones that are the same and the ones that are different. Different unit cells are going to be different polymorphs, and then you can initially start labeling. So this is polymorph 1, 2, 3, 4, 5, 6, etc., depending on how many unique unit cells that you found. You absolutely don't need a database or any references. If you have CSP, then you can match the unit cells you're finding to the CSP unit cell using like the ratio normalized to the volume, because of course CSP is going to be at 0 Kelvin. So you have to adjust for the volume change. But you can match to the CSP, and then the CSP will have some labeling or characterization of that polymorphic form, for example.
Okay, so this one is somewhat related. How can you be sure if a sample is enantiomer with ED?
Well, this is again moves into an area where I don't usually play. 'Cause, my focus is on disordered materials mainly, but you would have to almost certainly you would have to go through the dynamical pipeline if you want to look at the handedness of the molecular entity itself. You probably wouldn't pull that out from a rapid screening. The way that I would go about that is once I've got my unit cells identified, I would look at the unit cells that match or the same. So I'm collecting data on the same polymorph, then combine them together to get more complete data set. That data set I would then pass out through, well, I'll do initial structure solution and then pass it out through the dynamical pipeline through Yarn or OLEX to get the final handedness of the molecule itself. And then look at the various grains that I have in the material. If they're all the same polymorph, then that's clear, that's obvious. You can get it done very easily. If you've got multi-polymorphs in there, then it can get a little bit more interesting. But even so, you can still extract the handedness from each of the polymorphs that are in there.
Okay, and we'll just do one more just now, and we can maybe answer some of the others in the next question session if we have time. So when determining unit cells, do you use multiple or single images?
These are multi-images, yes, yes.
Okay, right. Shall we carry on, and we'll come back to
Oh,
questions a bit later on?
absolutely, we can. So automation and multi-grain analysis, as I think I've mentioned many times so far, I think that automation, integrated workflow flows, multi-grain analysis, working with a powder grain by grain, this is how you really leverage electron diffraction in an industrial environment. Without it, it's way too painful to be in industry. With it, it's basically trivial. Um, and as I mentioned before, the way that electron diffraction is set up, it really is set up for automation and high throughput. And I consider this powders grain by grain. If you take nothing else away from this webinar, think electron diffractometer. There's a powder diffractometer. It measures powders grain by grain. And it doesn't really matter where these grains or the powder comes from. You can take a drug product, something during production. You can then just render it into a powder and scatter it across a grid. These can be synthesis products. You could be looking with natural products, isolating them. Could be liquid based. You pipette on there. And one thing I saw recently, which really surprised me, are these nano manipulators, where people are doing very targeted crystallization, making specific nanocrystals, and then they can pick them up and drop them on the grid in particular locations. So it becomes easier to characterize which experiment has led to which particular nanocrystal and how successful it's been. And then the automation pathway, at least in the Chrysalis Pro pipeline, it steps through each grain automatically. It'll align the grain into the electron beam. It'll do the first pass. Does it diffract? Is it crystalline? Second pass through multi-grain, multi-images. It'll then extract the index solution. Then we can choose to combine data together for the same unit cells and go into connectivity. And if needs be, we can then go into absolute structure and look into placing protons and the rest of it. This is like a flow pipeline through the automation process itself. And in the next slide, this is a video. It's been speeded up quite substantially. Typically, we take a minute to 2 minutes per grain, but it just illustrates the point. This is stepping through a series of grains, and you can have thousands of grains that have been identified. It'll step through grain by grain, and you'll see the next one. It brings it into the center, aligns it to the beam, tilts the sample, makes sure it sits in the middle of the tilt. Does it diffract? It's got peaks. Then it collects multiple images. You'll see there are multiple images there when we have a diffraction grain. So so multiple images. They're combined together to enable us to index the material. So this is how it appears, speed it up a bit. Each grain takes about 1 to 2 minutes in reality. Although that we are actually speeding that up. I think it'll be definitely sub 1 minute, probably in the next few months or so. Each iteration of the automation algorithms is faster and faster. So what does that give us at the end of the day? We get a table of grains with their physical locations on the grid and their unit cell results. And attached to that will be the complete image data set if we want to go on and do connectivity or something more complex. So we then feed these unit cells into like a hierarchical cluster algorithm, and we generate a dendrogram. And this data is actually from the dehydration of trehalose hydrate. And you can see the multiple unit cells that are formed during that dehydration process. When they're the same color, at least the way this has been displayed here, they're considered to be similar unit cells. These data sets can be combined together to improve completeness. And then we can start looking at connectivity and finally moving on through a dynamical pipeline to get absolute structure. Now, we can often get the connectivity from a single grain. We don't necessarily have to do multiple grain combination, but it does give you higher precision and higher completeness if you approach it in that way. So just to illustrate that again, and then we'll move on to a different part of the story. But if we take this pharmaceutical powder, for example, and I put it in a powder diffractometer, this is the pattern that comes out. Now, I've worked in pharma so many years. Looking at that, I can recognize that we're dealing with lactose monohydrate for sure. just by looking at the data. But surprisingly, when I took that data through, a search match, I couldn't find what the API was. I couldn't find it at all. But if we put that on an electron diffraction system, sub-micrograms, nanograms of powder, we set up the automation process, steps through the grains, you know, 90% of them, or more than 90% in this case, are the excipient, lactose monohydrate. But then this one here, isolated in orange, this is the API. This is what we're interested in. And from that one grain, we get connectivity and a crystal structure. And I couldn't even see it in the powder pattern. So, this, it has tremendous power and utility that gives actionable data into the development pipeline and can really save you huge amounts of time in sorting out what's going on in your formulation process, or what polymorphs you're forming when you make the drug product, etc., etc. So we're approaching the end of our time here, and I just want to touch on a couple of routine applications. This is all published data. These are studies that we perform really on a daily basis. We do these types of analyses. And this first one, the first time I saw it, it just blew me away completely. I never imagined that such a thing would be possible. And this is a antihistamine a drug product, a solid dosage form. But the crystal form of the API in the drug product was really elusive. No one really knew exactly what the polymorph was, and there were IP concerns, all sorts of issues with what on earth was going on. Well, my colleagues just purchased this over the counter and then took it back to the lab, rendered it into a powder, spread the grains out. They measured probably 1,000 grains, I would say, a lot of it. And that 10% of the grains they identified was the crystalline API. And they had sufficient data to pass that through the dynamic pipeline that existed at that time. Early on, we only had YANA 2020 as the optional dynamic pipeline. Now we have it integrated with OLEX as well. And from that, not only were they able to get the unit cell, the crystal structure, the connectivity, but also the absolute handiness of the molecule itself. And to me, that was really a tremendous result. And seriously, I couldn't believe it the first time I saw it. But now we do this pretty much on a daily basis to try and work out what are the polymorphic forms in the drug product. Is there any IP infringement, reverse engineering, counterfeit analysis, or all of that is enabled and supported with electron diffraction. And one of the more obvious solutions is looking at where the proton exists when you have co-formers in the crystal. So when you have more than one molecular component in the crystals that you're growing, you want to try and identify where the proton is. Is it transferred from the API, or is it still attached to the API? In this particular example, in indomethacin nicotinamide, the proton is not transferred, so we're dealing with a co-crystal. This is the same molecular entity as indomethacin. It hasn't changed. In this particular co-crystal, again, the protons have not been transferred, so these are the same molecular entities. And in the final example here, the proton has been exchanged. Now we have a salt formed, and so the molecular entity of the API, the active, is actually changed at this point. So because of the way the agency characterizes and deals with salts and co-crystals, you really have to know what it is you're dealing with when you have co-formers in the crystal. And the last of the routine applications I'll show here, this is protein crystal structure determination. And again, I would have thought this would be impossible with electron diffraction. I don't work in this area at all, but some of my colleagues now do this routinely. And the way that the experimentation typically goes is they deposit the protein crystals in some mother liquor onto the grid. They blot away any excess liquid, plunge freeze into liquid ethane, which sits inside a liquid nitrogen bath, pull it out, blot off any excess again, and then transfer it directly on the cryo stage into the electron diffraction system. And surprisingly to me, we are able to get protein structures on a pretty much routine basis and screen for ligands attaching to particular target sites. Now, proteins are pretty big things. And so the unit cells that form in the crystal structures have huge lattice parameters. And this has basically two effects. One is we get a lot of diffraction peaks that sit right on top of each other, and a lot of diffraction peaks at low angles. So to help us observe that and characterize the data, one of the really beautiful things about electron diffractometer is that the detector sample distance is a virtual distance. So we can just change that in the software. We can pull the detector back, we can bring the detector in, so we can change the effective resolution of the measurement. And this enables us, by pulling the detector back, to pull apart these diffraction peaks quite clearly, to isolate them, and then quite cleanly derive the protein crystal structure looking for ligands and active sites. So post-measurement capabilities, I mean, electron diffraction in the industrial environment is definitely evolving. It's relatively new as an industrial tool, but things that are being added on a routine basis are post-measurement. So other things that we can do with the electron beam once we've collected the diffraction data. And then typically for this, we turn up the current. This is a lab 6 filament. Usually it works at about 100 microamps for the diffraction measurements. But if we want to do imaging or something like that, we can turn the power up and get much cleaner images. So single images, very high resolution. We can do tomography, rotating crystals around, looking at the electron transmission, and also energy dispersive XRF. This is a relatively new addition to the electron diffractometer. So we have an EDS channel. So we can look at the elemental makeup of each of these nanoparticles that we step through and study. Here's an example for an acetyl acetonate coordination complex, where it can be difficult. They're isostructural, and it can be difficult to identify whether we've got iron or gallium in the structure. But from an EDS spectra, it's clear and obvious. On this particular one, we have no gallium signal, we've got a strong iron signal, and then on the lower spectra, we have very strong gallium. So this allows us to really identify when we have key elements in the structure, what sort of materials we're actually dealing with. And that can be very useful for like reverse engineering, looking at contaminants. You've got a customer complaint sample with a black speck. What is it? Put it in the electron diffractometer. Let's have a look. We'll let you know what the black speck actually is. So we have another polling question, Fraser.
So the polling question, which I've just launched, somewhat similar answers to the first one, but slightly different question. So we want to know which part of the pharma development pipeline you think that electron diffraction is best suited. So you don't need to work in pharma to answer this one. And if you have another idea, then please let us know what that is. Okay, so it's looking so far very much like the R&D side of things is where most people see the benefits.
Yeah, yeah. Discovery, I think, is an untapped, area for electron diffraction with the high throughput. At the present, we can do like 2,000 to 3,000 experiments a week, but our target is to reduce that down to 1,000 a day over time. So we can do really supportive high throughput screening in the discovery and early development phase.
Okay, I think I will stop that there. We've got roughly half of the attendees have now answered. So I'll share those results for you. And Simon, you can continue when you're ready, but I'll just point out that I think your video is frozen.
Oh, do you want me to try and fix it, or shall I continue frozen? I think in the interest of time, I'll just continue.
I think just continue.
And yeah, yeah. Oh, the slides are frozen too. Oh, here we go. So we are definitely rapidly running out of time, and I just want to quickly cover on some of the new areas that we're looking at. So this is all very, very new stuff. Um, so I mentioned at the beginning, we're able to look at lipid nanoparticle delivery systems. With the huge growth in nanopharmaceutics, there is a lot, we get a lot of requests to look at individual nano delivery systems. So these are little particles, they can be like molecular entities, whatever, like a monoclonal antibody where you're going to stick some payload on the back end, or a lipid nanoparticle where people want to isolate and do diffraction on the individual delivery systems themselves. And this is an example. This is doxorubicin API encapsulated in a pegylated liposome. The reticule size is about 2 microns here, so that you can see these individual particles. They're only about 30 to 40 nanometers, but we can isolate those, and we can do electron diffraction from that. And to the best of my knowledge, this is the very first electron diffraction measurements on individual lipid nanoparticle delivery systems with an API inside. Now, this is rather complex, and at this point in time, I couldn't tell you how to really analyze that, but we're exporting the data into a total diffraction format, and we're doing some PDF analysis to try and understand or get a structural fingerprint, if you like. Is this lipid nanoparticle the same as I've made before? Does this pass the QC test? Can I get some basic characterization out from those materials? Another area is disordered systems. Again, we have many examples of this, but I just want to pick one that I can actually talk about. This is furfuryl acid. These are usually treated with UV radiation to form these ladder structures, and we do this processing in-house. And this is an example of an electron diffraction pattern. So this is a three-dimensional reciprocal space. It's called Evol 3D in the Christus Pro software. And instead of seeing the nice rays of well-defined diffraction peaks we would expect to see for a crystal, this one is very, very messy. Very broad, diffuse diffraction peaks. There's also a change in symmetry going on here somewhere. So there's multiple changes occurring due to the over-processing of the material. And this is something we typically see in APIs that have been milled down to small particle size, to increase manufacturability. We can then begin to analyze this to understand what's happening, how is it disordering, how is this going to affect dissolution rates, how's this going to affect bioavailability, what's my performance of my drug product like to be like with these high defect materials. We can again export the data in total diffraction or powder format, that's in green, and we can compare that to the pristine material. And you can see that there's some serious changes going on there in this material as it's disordering. And really the last example, and this is coming out of our work looking at amorphous solid dispersions. And as I mentioned before, this is something I'm doing this week in our lab in Tokyo, looking at amorphous solid dispersions, trying to get early indication of API phase separation. But this is a measurement on amorphous carbon. And yes, electron diffraction systems will measure amorphous systems. It's like any diffractometer, X-ray, neutron. We can look at amorphous systems, crystalline systems, liquids, gels, whatever needs to be studied. And this is exporting the amorphous carbon diffraction data, the three-dimensional reciprocal space data, as a powder pattern. Very beautiful, very clear. And also notice down here that the powder pattern is done by 4 degrees 2 theta. With this extremely short wavelength, 0.0251 angstroms, the whole diffraction is done very, very quickly. So the detector doesn't even need to move anywhere during that measurement. And then we can convert that to a total diffraction format. This is QS of Q minus 1. And this is really beautiful for pairwise distribution function analysis, so we can get a nice idea of the structure inside the material. And this is the approach that we're using to understand what's going on in amorphous solid dispersions, particularly risk analysis and risk prediction. So on that, I will move on to the summary. This is just basically going over what I've discussed already. So hopefully now I've made believers of you that electron diffraction has a place across the whole discovery and development pipeline. It can give you unique actionable data and can really shorten the whole development process substantially, because you can look at early materials, first solids. You don't need to scale up very often. You don't need to purify. You need nanograms of material. It works with powders. You don't have to grow singles. It's a tremendously powerful tool to have in the lab. So at the early stage, we're looking at synthesis processes, early polymorph screening, looking at the effects of impurity fields, how that generates all these metastable polymorphs. We can also look at crystal growth dynamics, even from solution out of the mother liquor, using the liquid cells. In the pre-formulation area, we've got all the polymorph screening based on unit cell, matching to CSP, for example, no scale-up required, very little purification, if any, required, identification unequivocally of salts and co-crystals. And then in the manufacturing side, we can characterize the API material within the drug product, looking for counterfeits, reverse engineering, IP challenges. And then in the formulation stage, we can look at individual lipid nanoparticles. And then back way early in discovery, we have this automated high throughput protein screening looking for ligands. So thank you again for staying with me throughout the presentation. And I think we have time probably for a couple more questions.
Yeah. So, do you want to maybe now try and turn your video off and on so people can see you again? Just
Uh,
for the questions, and while
okay,
you do that,
give me a second here.
I'll just yeah. So I'm going to ask the questions, which I think are most related to pharma, since this is a pharma webinar, and then if we have time, we'll do some of the others. So there's one with an upvote, which is, in what ways can electron diffraction be applied to study the interactions between drug molecules and excipients in a formulation?
Hold on a second, my video is going on. Could you repeat that question again? I got lost in my camera settings.
Yeah, okay. Yeah, just leave it if it's
It's
trouble.
completely locked me out now. No,
Yeah.
that's it.
So the question was, in what ways can electron diffraction be applied to study the interactions between drug molecules and excipients in a formulation?
Well, I think that that can be done in a number of ways. I mean, if the API agents are separate within their own grains as such, then can you hear my audio?
Yes, yes, you're fine. Yeah.
Okay, I just got a notice from the Zoom is now no longer responding. Okay. So if they're in separate grains, then of course, we'll measure them through grain by grain. And once we render them into a powder, we essentially break that relationship where they were within the drug product itself. But what we can see, is what the structures are and how many of each of these structures are in the drug product. That's if the regions are separate, they're separate crystals. When we turn them into a powder, they're going to be completely separate on the grid. But if the molecules are really interacting together, like with an amorphous solid dispersion, where the API molecule is dissolving into another excipient or interacting very strongly that way, then yes, we will be able to see that. Typically, when they go in, they probably won't be crystalline. They may be, but I think unlikely. So total diffraction. So from those individual grains where the API molecules interacted with the excipient molecule, if it's dissolved into a polymer matrix or something like that, the total diffraction will allow us to see, what's going on locally within that structure.
Okay, so let me just select another one. So on slide 28, what is the detection limit for ED in comparison to X-ray powder diffraction?
Yeah, that's actually, a great question and something we have thought about a lot. Um, and from one perspective, you only need to see one grain to identify something inside a drug product. But when you actually look at the statistics of it, I mean, how many thousands of grains would you have to measure in order to see something at 0.1%, in order to see a statistical representative number? And what limits it is your ability to collect, or the time available to collect, the number of grains. Now, it's much like an X-ray diffraction measurement. Your limit of detection, your sensitivity is going to be limited by how long you want to collect the data for. And it's the same in electron diffraction. It's limited by the number of particles you actually want to analyze. In the routine screens that we do, the detection limit is pretty much the same as X-ray diffraction, X-ray powder diffraction. So we say 1%, maybe 0.5%. If you want to measure multiple thousands of grains, then you can push that down, maybe zero point 0.2. But to get it to like 0.01% routinely, I think would be seriously challenging. You'd have to measure so many grains, you'd be spending weeks collecting data.
Okay, one last question before we finish up. So can electron diffraction analyze the structure of API, which is captured inside lipid nanoparticles?
Yes, that is the whole point of the analysis that we've been doing on lipid nanoparticles. That is possible. Um, of course, the diffraction pattern is made more complex because you see the lipid on the outside, so the shell, you get the diffraction from that as well. So it's a more complex arrangement. But if the shell is more amorphous, more disordered, then the diffraction peaks stand out quite clearly. And then the
Oh dear. Looks like we lost Simon, unfortunately. So thank you for joining us. Any of the questions that we weren't able to answer, we will follow up with email responses to those. And I'd just like to also paste some links into the chat for you. So if you are interested in any learning anything more about the products we've talked about today, or the other episodes in the webinar, the recordings are available on the webinar series page, which I've just posted. You'll also be able to sign up from that page if you haven't already for the final two episodes. The next episode will be on the 7th of July, I believe. Sorry, the 9th of July, and it will be at the same time. And our presenter will be Joshua Bucarsley. And the topic for that one will be battery materials. So that is taking a very different direction and hopefully be very interesting. So please do join us. Thank you again for joining us today. And thank you to Simon for the presentation. And we'll see you next time. Thank you very much.